Good morning, everyone. Solentum is delighted to be the sole sponsor for this week's conference. We're delighted to see such a great lineup of speakers, some of whom have already successfully demonstrated the platform that I'm going to talk to you about today. In terms of the scope of the presentation, I'm going to begin with pointing out some of the needs of the equipment in this area. I'm then going to go through the details of our particular platform from Solentum for single-cell cloning of clinical grade iPSCs. We're then going to go through process development using GMP compatible reagents. And this is where my colleague Camilla Dominghetti will present recent data on testing for best combinations of GMP compatible reagents, particularly in the lifting, dissociation and cloning steps. I'll then come in and just talk about some other key aspects for GMP implementation, particularly around single-use consumables, and then we'll go on and do a final summary. So first of all, the needs in this area. I took this diagram here from a recent paper. And really the sort of main message here is that the master cell bank, or MCB, should be produced under GMP conditions. I'll also repeatedly make the point throughout the talk that the translation to manufacturing needs to be borne in mind throughout, and you need to start implementing things in the development which can then be copied over or replicated in the manufacturing. Key to all of this, and I've seen this in numerous presentations from various iPSC cell therapy companies recently, is getting a clone or master cell bank. So consistent, high quality starting material is absolutely key for this area. So what are the typical steps for a process development group? I'll put this in the form of a workflow in a minute, but most customers will be doing gene editing of the iPSCs, either single edits or multiple edits. They'll then go through a QC procedure for getting the optimal clinical grade clone. They'll expand for banking, then freeze this down or cryopreservation. They'll do QC for release testing, and then we have our GMP starting material. And bear in mind, the single iPSC clone can be expanded ultimately to make hundreds of vials in the master cell bank and working cell banks. So putting this into our Solentum workflow, I'll describe the platform in a moment, but first of all, sort of notice the short timelines for doing this process. 19 to 25 days from basically the gene editing through to banking the clones. And this is a single round of cloning, I should emphasize, using the VIPS platform. But also look at this funnel down at the bottom. This gives you a sense of the number of plates, input clones you need to do with a gene editing project, and the various triaging at the different steps of clonality, expanding, and then confirming with sequencing or genotyping the particular edit is in place, and then banking down the clones of choice. So next, I'm just going to take you through our combination platform for single cell cloning of iPSCs. So let me take you through the VIPS instrument first of all. VIPS stands for Verified In Situ Plate Seeding. It's been a very successful commercial product for the last 3 or 4 years for our customers that are making cell lines for secreted biotherapeutics like antibodies and viral vectors using CHO and HEC. More recently, it's been specifically developed around iPSCs, which are very, very challenging in their own right. The main sort of facets of the instrument is, compared to, say, flow cytometry, this is a very low pressure system, cell-friendly I would call it, and we dispense cell droplets. So each cell is dispensed in a 20 nanoliter droplet, and it's dispensed into a well, but onto the bottom of a dry well, a dry surface. There's no liquid in there. and then we image through that droplet in the Z plane. We take about 20 images as a Z stack, and we confirm using artificial intelligence whether there is a single cell in there or a doublet or an aggregate, and that's fully automated, that confirmation of a single cell. Once we've got that single cell confirmed, we have a separate nozzle that fills the well with a predefined volume of media, usually about 100 microliters for an iPSC, and then we let that cell settle, and then we use that whole well image for evidence of clonality. And then uniquely to Solentim and the VIPs is that we use a combination of both the droplet image and the whole well image, and having a single cell confirmed in both of those pictures. That gives us our double lock of assurance. And then finally, we have the ability to put the representative images of the droplet and the whole well and the growing colony into a report that can be used with the regulator, for instance, as part of the IND process. Additional features I would like to point out are that the VIPs will usually sit in a biosafety cabinet, a class 2 containment. The time for seeding is approximately 10 minutes, and that includes both getting a single cell into the well, but also confirming with the software we actually have a single cell in there. And then the whole well image is taken approximately 1 hour after seeding to allow that cell to settle down to the bottom. This then is another feature on the VIPs, which is a dedicated iPSC confluency scan profile. Given that iPSCs are quite translucent, we've developed it using an AI-specific confluence detection, so we can find the cells, whether they're a single cell or a colony. So you can see, first and foremost over here on the right, we can identify that single cell with this green overlay on day 0. And then we see it on day 1 as a very sort of small colony of about sort of 4 or so cells, and then a slightly bigger colony. And then by the time we get to the day 10, you can see colonies easily highlighted there in green with the overlay. And importantly, we don't have any false detection around the edges that would corrupt or pollute that confluence value that we're giving. This then is the clonality report that I mentioned. This is a key element of the process where we capture the representative images in our report, and this would be for inclusion in an IND. And typically, going from the left to the right here, we'd start off with the droplet image from the VIPs. You can see the outline of the 20 nanoliter droplet with the cell in it. Then over here we've got the day 0 image, which is about 1 to 2 hours after seeding, and we're confirming the single cell. You can see it there on the edge of the well. And then we have typically the day 2 image, or the 24-hour image, where the cell has gone adherent and divided into 2 cells here. And then we could show over here on the far right images of the colony as it's growing. You see it around the edge of the well there, and you can put you can actually choose what images you want from the time course to include. So the second part and kind of key part of our combination platform is the enabling matrix. And note, I want to make the point here that by design, our system does not work with the vitronectin or matrigels. We specifically work with matriclone, or as it says here, the recombinant laminin 511. The key features of this is we actually use it in a soluble form. So we actually have it in the cell reservoir with the cells while we're seeding. It's a cell-friendly matrix which supports high viability and good growth of iPSCs. Importantly, when we get to manufacturing, it's defined, animal component free, based on a small recombinant fragment of laminin. Doesn't cause blockages in the nozzle when it's being dispensed. And it's importantly optically very clear for high image quality. You saw some of the images I showed a few moments ago. And then finally, there's a GMP grade source available, which you can then use in clinical manufacturing. And I'll talk to this later on when we talk about GMP consumables. Note as well that the process of using the matriclone in a soluble form throughout the process is a patent application by Solentim in the US. So next I have a short 3-minute video from my colleague Emma Morris, and she's going to illustrate to you how practically you would use VIPS and the matriclone in your lab and how easy that is. And then I'm going to hand over to Camilla, who's going to talk about how we've been doing work to test different combinations of GMP compatible reagents, particularly dissociation reagents.
Hello, I'm Emma and I'm going to be talking you through iPSC single cell seeding using Solentim's instrument VIPS. Before seeding, it is essential that your iPSC cell line is adapted to matriclone matrix. Matriclone is designed to support iPSC growth in single seeding workflows and is optically suited for automated imaging. We recommend growing your iPSCs on matriclone matrix for a minimum of 2 complete passages prior to single cell seeding. It is important that limiting dilution plates and serial dilution plates are always set up alongside your VIP seeded plates using the same cell suspension and growth medium. On the day of your seeding experiment, enough outgrowth medium to seed your 96 well plates with 100 microliters of media per well should be prepared. Outgrowth medium should consist of stem cell growth medium supplemented with ROCK inhibitor and matriclone. iPSCs for seeding should be selected from a 6-well culture plate which is approximately 50 to 60% confluent. The iPSC cell suspension should be titrated to a single cell suspension. VIPS can detect the difference between single cells and aggregates, so the presence of some aggregates is acceptable to preserve the viability. The viability of the iPSC cell suspension must be greater than 95% to proceed. Once seeding is complete, the iPSC confluency scan profile should be used to perform whole well imaging for all time points on the VIPS, limiting dilution and serial dilution plates set up. Whole well imaging should be carried out 1 hour post seeding, day 1 following seeding, day 4, day 7 and finally day 10. It is important that all VIPS, limiting dilution and serial dilution plates follow the same feeding routine throughout the outgrowth experiment. All plates should receive a full medium change on day 2 with stem cell growth medium supplemented with ROCK inhibitor. From day 4 through to day 10, all plates should receive a full medium change with stem cell growth medium alone. I hope you enjoyed this short video. Thank you for watching.
Thank you, Ian and Emma. Hello, my name is Camilla Dominigetti. I am the Laboratory Manager here at Solentim, now part of Advanced Instruments. Today I will be discussing with you some of our current research, and just to follow on from Ian's discussion earlier, our main focus has been investigating GMP compatible reagents and how we can implement these into our iPSC protocols and workflows, alongside using the Solentim instruments, the VIPS and the Cellmetric. So the main experimental study that I will be discussing with you is our investigation into GMP dissociation reagents. So we have 3 commercially available reagents that we have been investigating, and for the purposes of this study I have named them lifting reagent A, B and C. So the main aims for this study were to evaluate the reagent's ability to firstly dissociate the cells, secondly to monitor if they affect the cell viability, and finally, and probably most importantly, to monitor whether they affect the percentage seeding efficiency and therefore the percentage clonal outgrowth. So for this study, a iPSC cell line was grown into a 6-well tissue culture treated plate, and on the day of seeding, 3 wells were chosen based on the fact they were 40 to 50% confluent. The cells were lifted using each of the dissociation reagents and then diluted down to 9,500 cells per mil. We then measured their percentage viability and found that they were all greater than 95% viable, and we find this suitable for single cell seeding when using a VIPS instrument. As part of the VIPS seeding, we completed 5 96-well plates for each of the lifting reagents, so 15 plates in total, with a media fill of 100 microliters. The plates were then placed into the incubator, and after approximately 2 hours of settling time, the plates were scanned on the Cellmetrics software for whole well imaging. Further time points took place for Cellmetric scanning, and this was at day 1, 2, 4, 7 and at day 10. And just as a note, all reagents and medias remain constant throughout this experiment to remove any variability and to allow the data to be comparable. So here you can see some of the results from this data. So if you look at figure 1a, we initially investigated the percentage VIPS seeding efficiency. And you can see from the graph that lifting reagent C displayed the highest average percentage seeding efficiency with a value of 80%. And this was approximately 17% higher than lifting reagents A and B. And if you look over at figure 1b, we also investigated the percentage and number of cell aggregates across the plates. Again, this was taken as an average of the 5 plates for each lifting reagent, and we found that lifting reagent C gave the lowest number of cell aggregates with a value of 2.7% per plate. And again, this was considerably lower than lifting reagents A and B. When you put these two data sets together, it strongly indicates that lifting reagent C is the optimum lifting reagent for maintaining a single cell suspension when seeding with the VIPS instrument. We also investigated the percentage clonal outgrowth and whether this was affected by each of the lifting reagents. So you can see from figure 1c that again lifting reagent C provided the highest percentage clonal outgrowth with a value of 25%. And this was approximately 5% higher than lifting reagents A and B, taken as averages from the 5 plates. So just a few key points from this experimental study. We found that lifting reagent C provided the optimal results in relation to seeding efficiency and maintaining a single cell suspension, but also for percentage clonal outgrowth. It gave the fewest number of cell aggregates, enabling for the highest seeding efficiency, and it provided the highest percentage clonal outgrowth of the plates. A second experimental study that I will be elaborating on is our investigation into single cell seeding using the VIPS and a GMP outgrowth media which was supplemented with matriclone. The main aim of this study was to evaluate the highest percentage seeding efficiency and percentage clonal outgrowth we could achieve using the VIPS instrument. And for this study, the iPSC cell line was again grown onto a 6-well tissue culture treated plate. On the day of seeding, a well was chosen which was 40 to 50% confluent, and the cells were lifted and diluted to 9,500 cells per mil for single cell seeding using the VIPS. As part of this study, we seeded 5 96-well plates using this VIPS instrument, but alongside this we also seeded 5 plates using the traditional method of limiting dilution. The plates had a media fill of 100 microliters, and once they were single cell seeded, they were placed into the incubator at 5% CO2 and 37 degrees. We then imaged on the cell metric for whole well imaging at specific time points so that we can monitor the colony growth. These specific time points included at day 0, so approximately 2 hours after seeding, and then again at day 1, 2, 4, 7, and finally at day 10. You can see from the data here on figure 1D that the average percentage outgrowth of the plate was considerably higher when you use the VIPS instrument in comparison to limiting dilution. The combination of the VIPS instrument, the GMP compatible media supplemented with Matriclone, provided a percentage clonal outgrowth of 23.8% compared to the limiting dilution method, which had a percentage clonal outgrowth of 8.75%. The average number of clonal colonies per plate using the VIPS single-seeded system was 33, and we found very little variation across the 5 plates with a standard deviation of 1.89. So from this study, we have found that the combination of VIPS single seeding in combination with the GMP outgrowth media supplemented with Matriclone can provide us with a percentage clonal outgrowth of approximately 24%. This is 15% higher than comparing to the traditional methods using limiting dilution. Finally, I would just like to elaborate on some of our planned future iPSC experiments. So firstly, we would aim to further investigate lifting reagent C with the aim to reduce the concentration that we use alongside the incubation time to promote higher cell viability and therefore clonal outgrowth. We also aim to evaluate different GMP iPSC outgrowth medias, which could improve the colony growth, cell viability, and we can investigate the potential for weekend-free media changes. Finally, we aim to experiment with different GMP media supplements to improve single-cell survival and colony growth. Thank you very much, and I will pass you back to Ian.
Thanks very much, Camilla. I'd now like to cover some other considerations when you're planning for future GMP implementation. So Solentim are uniquely positioned to help support our customers and future customers with GMP planning and implementation. This is something I would point out that we cover under a non-disclosure agreement with prospects and customers, and it's something to bear in mind. In summary, we need to think about the areas listed below. So number 1 is control of the source materials. I'm going to go through this in some more detail in the following slides. So firstly, the matrix, and secondly, single-use consumables. But other things I'm not really going to cover today, but would be in our sort of deep dive discussions under NDA, are helping you with standard operating procedures to implement on your own site, maintain and develop your own SOPs. The importance of training and GAMP5 qualified training from our side, and proper training of all of your operators who will use the system. Considerations of the operating environment where the system will go. Typically these systems would be going into and out of manufacturing suites, so there'll be aspects of IQ, OQ. We need to consider airflow and accessibility to the instrument as well, and also cleaning of these instruments. You know, when you finished, what and how you clean these instruments down with. Firstly, let me talk about the matrix. This will be the transition point from using our research Matriclone in your process development stage through to swapping to the clinical grade laminin in the manufacturing process. So Salentim are the global distributor for GMP grade laminin 511, and we can provide you again, our customers under NDA, documentation on the source of that material, the cell line that makes it, and the provenance for that cell line, and also certificates of analysis and testing for that laminin. In order to procure this clinical grade material, customers must sign a supply agreement with us, and then the product will be drop shipped directly to you, the customer, from the manufacturing site in Japan. The other key part of the consumables is the need for a single-use flow path. So some iPSC customers that we interact with on evaluations already are pretty successful with flow cytometry, I have to say. Though this can work in a research and process development setting, it's not a suitable solution for manufacturing. You absolutely have to have a single-use disposable flow path in manufacturing. If sorting is key, and I've not seen a huge amount of it, but if it were key to your process, you could still use a flow cytometer for the enrichment step based on some sort of marker where you sort the population, but then you will put that population into the cell reservoir on the VIPS at the end and use the VIPS for the single cell cloning stage. So I'm very excited about this slide to present to you. So Salentim have been developing over most of the current year a new single-use cell reservoir, especially for our iPSC cell therapy customers. And these are currently starting to be tested with our existing early adopter iPSC customers and will be available for the VIPS in the new year. And these will come with a full certification of critical characteristics. So over here on the left, this is the original and current cell reservoir used on VIPS. It's made of a PEEK plastic and has a separate cell reservoir body and a separate nozzle which is attached to it. You can see that it's opaque, and typically it would be reusable, which is attractive in the cell line development community to rewash and reuse. But over here on the right, we have the new single-use cell reservoir for our iPSC cell therapy customers. So key differences, you can see it's a clear material. You can actually see into it. You could see the cell suspension, and you can visually validate the stirring flea is working and stirring the cells while they're in there. It's been sterility tested and gamma irradiated and comes in a sealed bag. And then just here where the laser pointer is, although in this photograph for demonstration purposes we're not holding it, were you to take this product out of its packaging, you would pick it up by this leading edge and attach it to the VIP. So you're only holding this one edge of the consumable and minimizing handling. And as I say, this comes with a full certification for this product ready for manufacturing. So finally, I'm just going to summarize briefly some of the things we've spoken about today. So hopefully I've made the point that we start with the end in mind. In other words, what you think about implementing and setting up in process development now, it would make sense if you think further ahead to manufacturing and implement in PD what you can then replicate in manufacturing. And that's both in terms of instrumentation, matrix, but also combinations of reagents like Camilla spoke about. You know, you can start testing and working with reagents that you know are GMP compatible. Efficiency, I mean, what we've tried to show with the VIPS is, and the aim here is to get more clonally derived colonies per plate. Now this is always going to be cell line dependent and edit dependent, but we've got plenty of levers to play with on the VIPs to develop and keep optimizing and tweaking that workflow to increase progressively the number of clones. So, for instance, that 1 hour time frame between the single cell droplet and confirming the whole well image, we found in some recent evaluations it's really important to stick to that 1 hour, because if you leave it to 2 hours, some of those clones have already started dividing, and although they were clonal, we can't prove with a double lock that they're clonal because the cell has already divided, and we've effectively lost some clonal wells we could have played with. So these are simple things just to improve upon to get more clones per plate. We showed the documentation process in the clonality report. This has been tremendously important with the regulator for CHO cells, and we believe it will be exactly the same for iPSC clonal cell banks, that you will need to show these pictures and have this documentation ready for your IND meetings with the regulator. And finally, just wanted to really highlight where Solentium are in this process, that we're very much we think at the leading edge of this technology and bringing some thought leadership to this process. We're working constantly on the biology in our own labs and on the workflow, and we would like to work with our customers and our early adopters to keep sharing this information and improving their processes. So it's almost like a sort of partnership process. And then finally, I had mentioned before about doing a deeper dive with customers on GMP. So I put my email address here. So if people do want to have a separate offline presentation and sign an NDA and find out and discuss more about the GMP manufacturing side and how we support you, please do drop me a line. So with that, I'll stop, and Camilla and I will now be available for questions. Thank you very much.